and cellular therapies here at Dana-Farber. She is a professor of medicine and the Levine Family Chair for Preventative Cancer Therapies. She received her MD from Stanford University and did her residency and fellowship at the Brigham and here at DFCI. Kathy has a long track record of working on CLL, identifying molecular drivers, and of course investigating the immunology of CLL. But then she also started another major program which focuses on personalized cancer vaccines, specifically on neoantigens, and she has been one of the major pioneers in this field. She had a landmark publication in 2017 in Nature demonstrating both feasibility and immunogenicity of personalized neoantigen vaccines, first in melanoma, and has since expanded this approach to a variety of other cancer types, including glioblastoma, which was featured in another Nature publication. Kathy has received many awards for her work. She's a fellow of the AACR Academy, but more recently, she has received a major international award, which is the prestigious Svedberg Prize, which is awarded in Stockholm, awarded annually by the King of Sweden on behalf of the Royal Swedish Academy of Sciences. And so Kathy is going to give us an update today on her work on cancer vaccines. Kathy, we look forward to your talk.
Kai, thank you so much for that introduction, and I'm regretting that I didn't pull in some photos of that time in Stockholm, but it was very, very fun. It's really great to see so many friendly faces who have kind of, traveled with me along this journey. I do want to revise one thing that Kai said, which is he said that I worked on CLL and then vaccines, and I think from today you'll see that the vaccines was kind of an underlying current all along, and maybe CLL was maybe the accidental. These are my disclosures. And I want to start here because I think, too often we wait to thank our family in the end. And actually, when we kind of take on these journeys, they really require, so much support. And I first and foremost, I want to acknowledge my family. This is my husband, Nir. He's been my life partner and really has been there from the very beginning as we kind of developed the story out together, and my parents as well, who have kind of seen us through all of these journeys, but as well as our kids who over the course of the time that we developed these neoantigen vaccines, they were always there kind of in the background certainly as a distraction, but certainly with a lot of love and fun. But so I think I do want to roundly acknowledge the support system that's been out there that has allowed us to do all the work that we do. So I think it's always important to ask ourselves, what is the key challenge? What are we really trying to address? And I think for all of us, we would acknowledge that we are roundly dealing with tumor heterogeneity, plasticity, and evolution. So it's this heterogeneity that's driving therapeutic resistance and the challenges that we face in treating cancer. And so this is kind of a redrawing of the 3 E's originally set out by Bob Schreiber, so elimination, equilibrium, escape, that it is this evolutionary aspect. Unfortunately, by the time we diagnose many of our patients with cancer, they've already escaped control. And so what's on the bottom part of this image is kind of the host immune system, because the cancers are not growing in a vacuum within our bodies. It's rather that there's a dialogue and a coevolution with the immune response. So unfortunately, by the time we diagnose the patients, there are already so many different mechanisms by which the cancer can evade immune surveillance and control. And this propensity to evolve was recognized very early on. So even in the 70s with Noel, at the first moment that we recognized that cancer was really a disease of somatic mutations, there was recognition that this was going to be a therapeutic conundrum. And he said, given all of this, immunotherapy becomes a leading candidate for the easiest means of destroying the remainder of the neoplastic clone. So I think this has been a challenge that has been recognized for a long time. Now, it's hard to believe that, because I think immunotherapy and cancer immunotherapy, this was not always the coolest therapy that's out there. But I think by now we recognize that immunotherapy is a pillar of cancer treatment. At the center of all of this activity is the T cells, and we recognize that T cells have so many different types of functions, and our immunotherapies are each trying to capitalize on those unique functions. And so that's why we see that there are so many different, diverse types of immunotherapy out there. But certainly for today, I'm going to talk about vaccines. And vaccines really stand apart from some of the other cancer immunotherapies in that they really provide an opportunity to expand T-cell responses and promote immunosurveillance and memory. So in this very complicated figure, what we're trying to convey is that in the red are those pre-existing anti-tumor T cells. So therapies like TIL therapies or checkpoint blockade are capitalizing on these pre-existing anti-tumor T cells and getting them to work directly or better. On the other hand, we have T cell gene therapy, where in the green we're trying to create de novo anti-tumor T cells. But vaccines uniquely have the ability to not only expand those T cells that are already existing, but also generate new responses as well. And I want to also say that I think the road toward cancer vaccines has been tough. We are more and more used to thinking about vaccines thanks to the pandemic. But I want to point out that infectious pathogens in cancer are definitely not the same. When we're talking about infectious disease, we're thinking primarily of prophylactic vaccines. This is in the setting where we're clearly dealing with foreign invaders, there's foreign antigens, it's highly immunogenic. The immune system is in a naive state to these antigens. In contrast, in cancer, we're dealing, as I said before, with that genomic heterogeneity. There is immune evasion going on. Because of the chronic antigen exposure, there's also chronic inflammation. There's this problem of T-cell exhaustion that many of us talk about. And as I mentioned before, by the time we diagnose patients with cancer, we're dealing with therapeutic vaccines. So how do we mount antigen-specific immune response when the cancer is already there? And so this field has been around for decades. There's really been a roller coaster in terms of development of cancer vaccines and whether or not we think it has clinical potential or not. But I think that, as I'm going to say in the remainder of the slides, I think we're really at an exciting time. So how did I get into all of this? And so a strong belief system for sure. But I think that you have to kind of think back again. I mentioned that this was cancer immunotherapy was really not popular when I was a postdoc in Jerry Ritz's lab. We were kind of in the corner there quietly working on our immunotherapy. And around the time that I was a postdoc, this rather influential article came out by luminaries in the field and this was from the NCI, and they reviewed their cancer vaccine trials, 440 patients, and basically they reported out that the objective response rate when they reviewed their experience was 2.6%. That is like a major bummer. So I think it's kind of reports like this that really turn people away from this field. Turns out that I'm an optimist and I am glass half full. Most of the time, I think folks in my lab can tell you that. But I think if you read this paper, you'll realize that it's the pitfalls at that time, the reagents that we had, but also many of the studies were single antigen targeting vaccines. So if you think about genomic heterogeneity of cancers, this was already going to be a problem. And if you even look a little bit farther and think, were there some glimmers of hope in that 2.6%? There was signal with more complex vaccines. So when we think about whole tumor cell vaccines where there were more antigens available, there was some signal there. So in this era where there was really not that much interest in cancer immunotherapy, you either studied melanoma, like many of our colleagues, or you went in the direction of stem cell transplant, and that was the path that I chose. I was introduced to allogeneic stem cell transplant as a medical intern at the Brigham Women's Hospital, and I just thought it was fascinating that you could have a normal donor immune system that could reconstitute in a patient with leukemia, and that there was an opportunity for that reconstituting immune system to, in essence, recognize the leukemia cells as should not be there and eliminate and eradicate them. Nicoletta Cherry, Katie Maurer, and I had the opportunity to write a review on the 30-year anniversary of Donald Thomas's Nobel laureate for clinical cancer research. And as we kind of said in this review, what was really interesting about stem cell transplant is that I think it really is the forebear of the modern cancer immunotherapy that we see today, because the concepts that we identified and are still active today in stem cell transplant is really the concepts that anti-tumor T cells are present and are important for response, that immunotherapy targets, their identification and going after them is the sharp point of the spear when we're dealing with cancer immunotherapy, that there's a conundrum of immune overactivation such as graft versus host disease, and these days we're thinking about CRS and autoimmunity. And to me, this is the ultimate combination therapy. So you're bringing many different modalities together and you get cure at the end of it. And so for my own research program, I've really focused on this concept of what are the immunologic targets that we are seeing. And so I think that this is what kind of brought us to neoantigens. And I think when we think about when these sorts of discoveries happen, really science is incremental. But there are moments of transformative technologies that allow you to leap forward and really get to the next level. And I think that at the time that I was emerging from my postdoc and starting my own career, there were many interesting technological things happening. The first was that there was new availability of next generation sequencing. There was also improvement in epitope prediction. So when I was a postdoc, we were really just only focused on HLA-A2. And suddenly with all the additional neural network models that were being generated, it was possible to predict across HLA alleles with greater accuracy. And I think it's really important to emphasize that around this time, there was also the new availability of immune checkpoint blockade with FDA approvals that really roundly changed the treatment landscape of patients. And so we became interested in neoantigens. So I think in terms of immune checkpoint blockade, I think the analogy that I like to make there is just if you make the Star Wars Death Star sort of analogy, is that suddenly that there was a way to bring down the Raider shield. I call it the Obi-Wan Kenobi moment. And suddenly it's possible to get targets and the fighters to come in. And so this is where kind of coming in and thinking about the specific antigen target becomes important. So what was the concept? What did next generation sequencing afford us the opportunity? Well, in the same way that sequencing allowed us to identify DNA mutations and in turn discover cancer drivers and pathways that were part and parcel of driving cancer, this also gave us a new therapeutic opportunity, because certainly some of those mutations could be transcribed, translated to have altered peptides, and at least some of those peptides could have the capacity to be processed and presented on the surface of a tumor cell where there could be recognition by a T cell. So this idea of a neoantigen that could arise from somatic mutation, well, that was not a new idea. That had been already conceptualized and postulated decades before. And it was always thought to be the optimal tumor antigen because coming from a somatic mutation, exquisite tumor-specific expression. And since these are somatic events akin to pathogens, there would be a lack of central tolerance, so high immunogenicity. And this is really a different place than where we had been in the previous moment in time, which is focusing on shared overexpressed tumor self-antigens with variable tumor-specific expression and variable immunogenicity. But what sequencing changed was suddenly there was a ability to discover those tumor neoantigens on an individual basis in a reliable fashion. So suddenly you could generate a pipeline, for example, and through exome sequencing identify those mutations and use RNA sequencing to confirm that those mutations were expressed. And then you could implement a prediction model for that incorporated HLA binding, so then you could come up with your candidate antigens. And so For me, this was really not a huge leap of faith, because in the area of stem cell transplant, we had already had evidence to support that such a genomic change could be immunogenic. One line of evidence that we had, our group had already started working on whole tumor cell vaccines in CLL, so that's where the CLL part comes in. We had already shown that with these complex vaccines that we could generate CLL-specific immune responses, and when we started to look underneath what those responses were against, it appeared to be neoantigens. We had already completed a study in patients with CML who had, by then, patients with CML were being treated with Gleevec. We understood that there was mutated BCR-ABL that was emerging as a resistance mechanism in patients with CML. And so we were able to scan the known highly recurrent mutated BCR-ABL mutations out there and identify which of them were predicted to be immunogenic. Turns out that in patients that went through stem cell transplant who had the mutation and the right HLA context, we could, in the setting of their developing remission, find that there, in fact, was T-cell responses to that mutated target, and that these responses came up only after transplant, and as I said, in the setting of remission. And finally, it had long been understood through lots of other orthogonal approaches that molecular changes could be immunogenic. So in stem cell transplant, we talk about graft versus host disease. We talk about single nucleotide polymorphisms, so genetic disparity between donor and recipient, and that at least some of them can have immunologic sequelae. They can be recognized. These are called minor histocompatibility antigens. And it was already known that such minor histocompatibility antigens could generate different types of graft versus host disease and maybe even generate graft versus leukemia responses. So it wasn't a huge leap of logic to think that mutations could also generate such responses as well. And so by the time we were starting to generate our pipelines, many others in the field were also working on this idea as well. And in general, there was growing understanding that neoantigens did in fact appear to be effective tumor rejection antigens. And just to review that a little bit, there were generally 3 lines of evidence that supported this idea. So the first type of level of evidence was that if neoantigens are as immunogenic as we think, then shouldn't it be that neoantigen load will be associated with better clinical outcome? And so there was certainly many different manuscripts that kind of looked at TCGA or specific diseases or following immune checkpoint that more or less had like some sort of Kaplan-Meier plot like this. A second level of evidence was this idea that if neoantigens are so immunogenic and associated with tumor rejection, shouldn't it be that we can find settings where there is evidence of immune control, and shouldn't we find evidence of expansion of neoantigen-specific T cells? And so our group certainly looked at this following transplant, but other groups looked at this after TIL therapy or after checkpoint. And then ultimately, if this is really a bona fide good tumor antigen, then shouldn't we be able to kill tumor cells in vitro and in vivo? And very nice human and mouse data from Bob Schreiber from the group in Genentech and also from Rosenberg demonstrated that neoantigen-specific CTLs could in fact lyse and kill tumor cells. So all in all, this was an exciting time to kind of think about this new paradigm shift that we could actually get to tumor specificity, not hit normal tissue, and really confront tumor in a multi-pronged fashion, so dealing head-on with heterogeneity. And a schema that our team has frequently thought about is whether or not we can use cancer vaccines as a way to steer the immune response. So if we already have immune checkpoint blockade as a way to release the brakes, and we have good ways to accelerate the response with the adjuvant, can we not steer the response to specifically expand and broaden the T cell repertoire by inducing tumor-specific T cells? So a question that our group asked ourselves is, well, if we can identify neoantigens, well, can we actually generate neoantigen targeting vaccines that can actually stimulate tumor immunity in human patients. And so as a very optimistic young investigator, I laid this all out in one of my first R01s, and I said, look, we're going to go from DNA sequence to actually identification of targets and demonstrate all the immunogenicity. And basically, the reviewer basically said, there's a reservation about the translatability of this work, considering the sophisticated processes of neoantigen identification and characterization. And so my family will tell you that, there was only moderate enthusiasm for this, and my family will tell you that I'm an optimist, but I also have a little bit of a contrarian streak. So the more you say I can't, So we set out to kind of create like a clinical program, and this was a early conception that Ed, who's in the audience, Ed, Nir, and I kind of sat down and put this together, and we thought, why can we not start from tumor, go do it, sequence it, perform the HLA typing, come up with a manufacture process. You have to look at the date. So at that time, we went with synthetic long peptides because there was already a lot of great human data that was out there that demonstrated that you could put even up to 15 peptides together without any autoimmune sort of sequelae. We went with long peptides, so 30 MERS, 20, 30 MERS rather than 8, 9, 10 MERS because of also great data from Case Malif and others to demonstrate that long peptides would require a professional antigen presenting cell to take it up and then mobilize coordinated multiple arms of immunity to bring together a robust immune response. We were interested in putting this together with a strong adjuvant like poly-ICLC and think about the opportunity of combining this together with checkpoint blockade. And I don't want to go through a lot of published work, only to say that our initial studies, as Kai mentioned, were in high-risk melanoma. We co-published this at the same time as Ugur Sahin. I don't think it's by chance that we both selected high-risk melanoma as the first setting, given that this is a high mutation burden tumor and that there was a nice adjuvant setting to go after. This was 2 years after Jerry Lynette and BHS Coreno reported out their dendritic cell vaccine, also using a computational approach to identify targets and also showed nice responses there. And then, as Kai also mentioned, we also worked in newly diagnosed GBM. This is together with Darren Keskin and also David Reardon and the team, and this was co-published at the same time with a consortium German group. I think the overall take-home from these studies is certainly safe and feasible. It's highly immunogenic. There, across the studies, there is highly promising combination with immune checkpoint blockade. And I think among our most exciting results were 2 patients that Patrick Ott treated, where on our first study, where they did have advanced melanoma, received the Neovax, so with the stage 4 disease, it wasn't a surprise that both recurred after the vaccine. But what was surprising is that they both went into complete clinical response within 12 weeks when their treating physician started them on anti-PD-1 therapy. And this, at the time that we reported this was a couple months out, but by now it's more than 5 years. Both patients are off therapy and doing well. And this is just imaging to demonstrate these lesions that went away 12 weeks after initiation of anti-PD-1 therapy. I think, as time has kind of gone on, we and others have been able to kind of access our vaccine studies and really find some really interesting and promising, encouraging short and long-term responses. And so just to kind of go through this, because we had our an original vaccine study in 2017, we were able, and this was an investigator-initiated trial, we were able to keep in contact with our patients and look and take a look-see to look at their responses even 3 or 4 years afterwards. One of the things we were able to do was, and this was actually work that we did together with Kai in generating class 2 tetramers, we were able to pull out those neoantigen-specific T cells at the single-cell level, look at their transcriptional programs, and we could see that there was actually a time-resolved cascade of responses in the patients as time went along. So going initially from a naive response to cytotoxic, then contraction, and ultimately a memory response, which is, of course, what we hope to generate with a vaccine. We were also able to look at the TCR clonotype profile. So the TCR gives a molecular barcode for these T cells and can kind of give you a sense of kind of the architecture and of the T cell response. And so if you follow this patient out, where each color represents the first time we see a clone, you can see that with the priming phase, you see red that persists across time, across priming and then boosting, but we see more colors as we shift to the right. And this really tells us that there is diversification of those T cells against the same neoantigen, but also persistence. And on the subject of persistence, when we looked functionally at blood that we collected from our patients close to 4 years later, we could still, these are triplicate evaluations on ELISpot, which looks at interferon gamma secretion. So if we look against all the different immunogens that were in the vaccine, we still see strong interferon gamma responses even close to 4 years later. So certainly functional, persisting, circulating. And finally, because we're using TCRs as a barcode, we can also track these neoantigen-specific T cells across different tissue sites. So I already mentioned to you one of the patients who had gotten vaccine, then recurred, and then got the anti-PD-1 therapy. When we looked at their blood, so if you look at the green and the orange circles, you can see at the single cell, looking at their single cell TCR profiles, there was a lot of overlap across those several weeks. So that's already kind of described in the middle panel here. But we also had available to, and we could already, the red dots indicate those TCRs that are definitively neoantigen specific. But because we also had the tumor specimen at the time that the patient recurred, we could use this molecular information and ask, did any of those neoantigen-specific T cells make it to the site of the tumor? And in fact, you can see that this is overlapping as well. And so we can begin to speculate that maybe the reason why anti-PD-1 worked so rapidly in this patient is because you already had those neoantigen-specific T cells, so soldiers at the site of tumor that were ready to go to work when the anti-PD-1 hit. We're not the only ones, this is not the only study where we've seen tracking of neoantigens T cells to the site of tumor. In work that we did in glioblastoma, we also had an opportunity to look at TILs in a patient with GBM treated with a vaccine at the time of recurrence, so after the vaccine. And we were able to compare those TCR profiles with in vitro stimulated T cells against the neoantigen. So we knew exactly which TCR sequences were related to recognition of the neoantigen. And what was remarkable is that we could see that there was overlap between those in vitro stimulated T cells and what was directly infiltrating in the tumor. And when we actually reconstructed those TCRs, we could confirm that some of those TCRs were directly recognizing the vaccine immunogen. So again, telling us that the vaccine generated T cells that were able to track, despite the blood-brain barrier, to the site of tumor. Recently, Vinod Balachandran and Memorial Sloan Kettering had very exciting data coming from a personalized vaccine for pancreatic cancer. Here they used an mRNA vaccine, and not all patients responded, but 50% did, which is already amazing for pancreatic cancer. And then those that did respond actually had a very excellent clinical response. But I think what was also exciting about their study is that there was support for immunosurveillance and tumor elimination at the site of recurrence by vaccine-expanded neoantigen-directed T cells. So this was a patient where they suspected that there was a recurrence. And by biopsy, they could identify that there was all these vaccine-expanded T cell clones present at that biopsy site. They couldn't find any evidence grossly of tumor, but when they molecularly performed an assay to detect the mutation associated with the tumor, they actually could find it. So the notion is that that probably the tumor did start to recur, but the T cells were present to eliminate it. One final point of evidence that collectively we've been looking at is this idea of epitope spreading following neoantigen targeting vaccines. So if we in fact have on-tumor targeting, then that cytolysis of that tumor will release their contents to be taken up in a secondary wave of immunity, and there may be new immune responses that were not originally in the vaccine itself. In fact, we were able to detect this was the case in some of our melanoma patients, where by week 16, we could already find responses against another mutated epitope, and that persisted. And in another study performed by Neon Therapeutics, they were able to also look at epitope spreading in their larger set of melanoma patients. Those that had epitope spreading had better clinical response than those that didn't. So altogether, over the past couple years, there have been a series of studies, whether it's using synthetic long peptides or by RNA vaccines. And I think consistently over time, we are seeing these very encouraging results at the level of correlative science, where we're beginning to see where we've seen complete responses to anti-PD-1 therapy after vaccine, decreased recurrences, tracking of these neoantigen-specific vaccine-induced T cells to the site of tumor, epitope spreading, lack of recurrences and immune responders after adjuvant vaccine. But really what's been very exciting over the past year is that we, for the first time, have a randomized open-label phase 2 trial that has been reported out in melanoma with clinical benefit. So this is Jeffrey Weber's work together with the Merck Moderna team, and they've been able to see this type of benefit in patients who receive vaccine and Pembro over Pembro alone. And so I would say that this is a super exciting time. I have fingers crossed, because now we're entering into a new era where there are planned or starting soon studies of hundreds and even thousands of patients, primarily in the adjuvant setting. So I think in the next couple of years, we're going to hopefully have a readout of these studies, and let's see where that takes us. So with all this activity, like, are we done? Do we have more to do, or what do we think? And so certainly we've been very well recognized for the work that we've been doing, and yet we are ever more sort of cognizant of the challenges that we have. And this is a nice figure that Max Sellers, together with Ed Fritsch, pulled together. Ed really loves the Golden Gate Bridge, and so this is kind of our rendition thereof, and we kind of put on the pillars that we have to kind of really address. So what are the key cellular players that we want to bring in? What are the antigens that we want to target? Delivery remains an active problem and challenge, and then what are the co-therapies? But we're really quite cognizant of the troubled waters that we're trying to bridge across. And so our general approach, so when I say our approach, this is kind of our larger personalized cancer vaccine team. We've kind of conceived of this bedside to bench and back again type of effort, where we really think about the clinical trial and how that gives us opportunities to think from where we've started. What are the new opportunities that we have? What are additional trials that we can address? But these type of very intensive trials also give us opportunities to collect biospecimens, and so we can entertain disease-specific research, and we are always trying to think about new capabilities so that we can do better with the human biospecimens that we have. And so just to give you a little bit of flavor of these three general areas in terms of trials, we have had ongoing phase 1 clinical trial efforts, and I just feel so blessed to be at our institute where we get to work with so many talented clinical investigators. Each disease type presents us with novel opportunities that we can kind of dig into one or another aspect. I'm just going to highlight one study that we recently completed with David Braun, who's now a junior faculty member at Yale New Haven, and this was done together with Tony Chuari. And this was really trying to think about high-risk adjuvant clear cell renal cell carcinoma so stage 3, 4 patients who went through total nephrectomy that had no evidence of disease after surgery. And we again used our kind of sequencing-based, genomic-based, prediction-based approach to identify targets. The synthetic long peptides were synthesized, and then we administered the vaccine together with poly-ICLC. There was a subset of patients who also received subcutaneous ipilimumab with the idea that maybe that would enhance priming. The vaccines were given in a prime boost schedule, like our other studies, so priming to increase antigen exposure up front, and then boosting to stimulate immunologic memory, and lots of blood collection. So in this group of patients, we really did see lots of promising results. So first, highly feasible, paucity of recurrences. So this is very cool. This is like 40 months from first vaccination. There were no dose-limiting toxicities. There was only one death, but it was, unfortunately, but it was unrelated to treatment or renal cell, but rather, unfortunately, to mental health issues. We saw robust immune responses, and including against neoantigens in known drivers of RCC. So, for example, this was a patient who had a mutation in BHL, and you can see this very strong immune response that was generated by week 16. And when you look across the patients, quite a few of them had mutations in drivers for which we could elicit immune responses. If you follow the red line, we could track over weeks to months, and we could see persistence of those vaccine-associated immune responses. But I think most importantly, we could detect not only responses against the vaccine immunogen, but against autologous tumor. And so I think that's actually what we want to get to. So we're certainly very excited about this. In terms of our disease-related research, this has really spawned efforts in my group to take a deep dive into the tumor microenvironment. I think we do need to understand the hostile environment that our vaccines are going into. One of the challenges of studying the native tumor microenvironment is the relative paucity of tumor-specific T cells that are there. But fortunately, we have available to us single-cell technologies right now that allow us to look at phenotype and integrate this with T-cell receptor information. And in addition to that, there's the possibility of trying to look and understand what the antigen specificity of those T cells are. So I think this is a opportunity to really kind of dig a little deeper. I don't want to dwell too much on published work, but Giacomo Oliveira and my group really figured out all the kind of technological sort of hurdles to get there. I just want to say that in terms of looking at the local microenvironment, we have, for example, been able to look in RCC where we can see that with greater stage of disease, we do see very active this kind of circuit of T cell exhaustion together with anti-inflammatory M2-like macrophages. In a disease like chronic lymphocytic leukemia, a blood malignancy, where we do know that there is sometimes a precursor lesion, so monoclonal B-cell lymphocytosis, when we kind of go into a single-cell dive, we can see that those immune dysregulation is happening even at that precursor stage. So this is already it's an early change that happens in that setting that. And it kind of underscores the challenges that we have to face when we send our vaccines in. For addressing kind of that idea of linking tumor specificity of the T cells, their state, and the antigen specificity, one of the approaches that Giacomo Oliveira and my group has done is actually come up with a way to link together hundreds of TCRs and testing them against hundreds to thousands of different antigens. And really, the way he's been able to do that is to miniaturize and parallelize T cell cloning and expression in normal donor T cells, and he's come up with an approach of combinatorial dye labeling so that you can put multiple single TCR expressing cells in one tube, and then through gating test against different targets. So it could be testing against a tumor or against the APC that is pulsed with antigen, whether you find it through immunopeptidome analysis, through prediction, or through known pools. And this is just an example of one of the different TCRs that were interrogated. This one was deemed tumor-specific because on the basis of 41BB or CD137 staining, you can see that there's reactivity against the autologous melanoma or interferon-stimulated melanoma, but not against autologous B cells or immortalized B cells. And in doing so, we've started to look at the TIL landscape of melanoma, where we can already identify that there's tumor specificity seems to reside in, seems to have certain sort of cell states. So the exhausted cells within the native immune environment in melanoma seems to harbor more tumor-specific T cells, whereas the viral specificity seems to be more in the memory region. We've been able to look at this not only in melanoma, but we've had the good fortune to work with Ravi Ubaluri and also start to look at this in head and neck cancer as well as in RCC. I think I will skip this. Finally, in terms of developing new kind of capabilities in terms of trying to maximize what we can learn from our human samples, we are more and more beginning to look at spatial cellular relationships, especially in the neovax setting. So if we, for example, have a clonotype for which we know the antigen specificity of, where does it kind of sit within the tumor microenvironment? What is the localization of those T cells, whether they're exhausted or non-exhausted memory? What crowd do the T cells that we're interested in, what crowd do those T cells run in? And so we've partnered together with Fei Chen at the Broad Institute to develop together SlideSeq, and together with Ken Levac and Shuchang Li at our institute in the Translational Immunogenomics Lab, we've worked together to create Slide TCR-seq, and more recently another approach that's defined single cell called Slide Tags. This is just one example of kind of a interesting sample that we worked on together with Charles Yoon, and this was a melanoma sample for which we were able to examine multiple regions of interest. This melanoma sample had 2 lobes, so here depicted in the blue and the orange, and what was so interesting is that there was actually a T-cell clone that seemed to reside specifically in lobe 2, in the orange region, not the blue. And when we started to ask, well, okay, does that T-cell, are there kind of what is kind of the monocyte macrophage interactions that we might see around that T cell clone? We saw enrichment for CXCL10, which we know is a T cell chemoattractant, and in relationship to tumor, there was a downregulation of MGST1 that we know is associated with cancer immunotherapy response. So I think these are the type of analyses that we're interested in kind of taking a deeper look at, especially in relationship to vaccination. So in terms of our ongoing work, personal antigen targeting does address heterogeneity. The current series of clinical and translational results are encouraging, and we've been fortunate to have strong cooperation with our motivated patients. Understanding the TME emerges as one of the foremost challenges to unlock, and at some level when we kind of do our sort of TIL analysis, I kind of think of it as kind of tumor immune cell archaeology. You see kind of evidence of past T cells that kind of came in that had the right specificity but became completely exhausted and now needs a little bit more fuel to kind of get back into the action. As we start to go from tumor to tumor, we do see similarities, yet stark differences across diverse tumor types. And I would say that there remains major opportunities for novel tumor antigen discovery and prediction and for novel delivery approaches. So can our vaccine approaches overcome the exhaustion that we observe in tumor-specific T cells in the native tumor microenvironment? Our vision is getting to a cure, and I think that I think one of the best parts of my job is working with young people and trying to develop kind of ambitious projects to forge forward. I've actually had this wonderful correspondence with Eleanor, who's 8, who kind of likes to give me advice on how we should kind of forge our next vaccines. So. She thinks, I think I have a cure for cancer, and she's kind of, and then in the end she says, I also think that you should get it through a shot. So love Eleanor. So this is kind of an ongoing dialogue. But, I think as we start to think forward, what are the lessons we've learned so far, and how can cancer vaccines fit in with the cancer treatment schema? And so certainly what I've talked to you, most of us are kind of really clinically addressing advanced metastatic disease, but we know that this is a very high bar for immunotherapy on its own to address, and at least in the vaccine realm, we really have to think smartly about the combinations that we need to actually get there. I think the most promising results we've seen so far has been in the prevention of relapse, so the adjuvant setting, and I think that we already know that if we can either administer in early disease or in settings of MRD, that's a very optimal place to go. And certainly, thinking forward, we are I think as a community, we're interested in cancer prevention, but this is going to require innovation in the way that we give the vaccines so that they're safe, cost-effective, and can be given to large populations of people so that we'll be able to see signal. But certainly an area of interest because of the potential for long-term impact on morbidity and mortality. I think, and yes, cost and time end up being very big considerations. I think as we kind of, my hope is that as we move forward that we can not only, for example, genomically characterize the tumor, but we can get a sense of the immune microenvironment so that we know what is the best combination of different approaches that can be given to our patients and in what sequence. And in the end, I really think that we're not too far in the future where we should be able to go to the clinic and just say, let's order me up a personal cancer vaccine, and it should be available to us within a few weeks. So that's the end of my presentation. I do want to thank all the votes of confidence over the years with the funding. This was a picture we took together with the Sjöberg Family Foundation. So it was a very, very lovely event in Stockholm, and it was very amazing to meet the family of the original donor who made this prize possible. I also want to take the opportunity just to acknowledge that really the Brain Trust for kind of our program has really been, as I mentioned before, not only Nir, but Ed Fritsch and also Patrick Ott, I think from the very early days, going to the FDA and all of this. This is, we've been down this road for quite some time, but I also want to take the time to acknowledge, all the amazing folks through my training period, starting from Marshall Wolf. I think many of you can say the same. Marshall, Margaret, Joe Anton, folks in Brigham Hematology, but also very much so our transplant crew, so Ted Allier and Rob Seufer, and immunologists in our community that helped shape some of the thoughts that kind of went forward to formulating our vision. Jerry Ritz, for sure, he was my mentor as a postdoc, continues to be a friend and a mentor. And then over the years, a lot of our work has been possible because of partnership together with the Broad. And so many fabulous folks over there, ranging from the sequencing and the genomic facility, computation, and proteomic crew has enabled us to do our work. We've had collaborations across Harvard, the clinical investigators, the folks that in our vaccine program who really make it happen, and so many other people. And I think I just want to say that at Dana-Farber especially, because we have the full circle, so the pharmacy folks, cell therapy, the clinical investigators, the CRCs, and then going into the lab has really made this type of work possible. So thank you for your attention.